Hello and welcome back once again to more human physiology. So this time we are going to start focusing in on some organic molecules. So this should be a fairly short video. We just want to kind of give ourselves a preamble into how organic molecules, especially those relevant to us in human physiology, tend to be put together and how they themselves interact with the water that we discussed last time. So we've talked about how organic molecules before, how kind of their defining features that they contain both carbon and hydrogen. Those that are essential to human function tend to come in four basic varieties: carbohydrates, lipids, proteins, and nucleic acids. The last video of this chapter will cover all four of them together, but for this video, we just want to kind of get an appreciation for how they're all put together and kind of what they have in common. So all of these molecules are very large molecules called polymers. So a polymer is a very large and very long molecule that is made up of individual repeating units called monomers. So we talked about monomers before. So monomers are put together to form polymers by repeated dehydration synthesis reactions. So remember how we talked about that before? So if you have two monomers and you perform a dehydration synthesis reaction to form a chemical bond between the two monomers, now you have what you might call a dimer. So a monomer is one, two of them together is a dimer, and then if you do another dehydration synthesis, you would have a trimer, so three monomers stuck together by a total of two covalent bonds. And then you can just keep building and building and building and building this polymer and make it as long as you want by incorporating as many monomers as you want. So we can use repeated dehydration synthesis to make a polymer that is very, very long. Or alternatively, if we already have a polymer and we want to shorten it or break it down completely, we can use repeated hydrolysis reactions to shorten it and break it down by breaking those covalent bonds. So in order for us to really go further with this and to understand those four different types of macromolecules we mentioned, we need to kind of wrap our heads around the chemistry of carbon itself. So if you will recall earlier in the chapter, we talked about how carbon is kind of a weird example. It has 4 valence electrons. So it's not exactly obvious in terms of how it's going to fulfill its octet requirement. You could almost see it either gaining 4 electrons or losing 4 electrons. It could do kind of either or. So it turns out both of those are going to be kind of hard to accomplish. So carbon is very much not a good candidate for forming ionic bonds. As it turns out, it is going to form covalent bonds. And every carbon atom is going to want to form a total of 4 covalent bonds, meaning each of its 4 valence electrons is going to participate in a shared covalent bond, meaning it should end up having a total of 8 shared electrons per carbon atom. So what this really means is that carbon can covalently bind to up to 4 other atoms simultaneously, and that includes other carbon atoms. So carbon is extremely versatile in terms of what kinds of molecules and what kinds of structures it can form. A very common one that we'll see here is the formation of what's called a carbon skeleton. So essentially you form a polymer of carbon atoms joined together one right after another. So in this particular case, we have a total of 7 carbon atoms. And then you will notice for each carbon atom, they have what we will call vacancies. The carbons on the end have 3 vacancies, and then the ones on the inside have 2 vacancies. So if we count them up here, we have a total of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 different vacancies. So for this carbon skeleton, we can attach 16 other atoms in a variety of different combinations, and you can kind of start to see how organic molecules in biology can be very versatile in terms of the types of things that we can build. So the simplest and kind of, I guess, laziest way, if you want to put it that way, to fill these vacancies is to just attach a whole bunch of hydrogen atoms, right? So in this picture, all 16 vacancies are filled with hydrogen atoms. Well, this type of molecule that we've made here, because each one of those carbon to hydrogen bonds is of the nonpolar variety, we have built ourselves a molecule that is highly, 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 highly hydrophobic. So this is actually not octane that we are looking at here. This is actually heptane, which is very similar. So if we were looking at octane, which it would be octane if it just had one more carbon and two more hydrogens. So something like octane is extremely hydrophobic because there is nothing about this molecule that water is going to care much for. So there's no electrical charges, there's no polarity, there's nothing that water is going to like to bind to. So this is a extremely hydrophobic molecule. It's going to want nothing to do with water. So what we can do instead of adding hydrogen ions is we can instead add a number of what are called functional groups. So a functional group is just a very small chemical structure. It's not always just going to be hydrogen. It's a very small chemical structure that can attach to a carbon atom and impart to it special chemical properties to the larger organic molecule as a whole. So this list here is by no means exhaustive. These are not the only functional groups, but these are some of the more common ones. So the first one you'll notice is called a hydroxyl group. Hydroxyl groups are actually polar. So the idea here is that if you go back to this hydrocarbon molecule here and you start replacing some of those hydrogen atoms with hydroxyl groups, well, that molecule, as you add more and more OH groups, is going to become more and more polar because hydroxyl groups are polar. So they impart polarity and hydrophilicity to the molecule of which they are a part. Carboxyl groups are also polar, but the thing that they do is they have acidic properties. So organic molecules that have a lot of these carboxyl groups have a tendency to act as acids. Amino groups are also polar, but they do kind of the opposite thing that carboxyl groups do. They tend to act as bases. Methyl groups are very nonpolar. You're not going to see these very often in biology, but you do see them sometimes. So they are going to be nonpolar because the carbon-hydrogen bond is a nonpolar bond. And then finally, the phosphate group is also very polar. It tends to be electrically charged, and you're going to tend to find these in certain types of lipids and nucleic acids. And we will talk about all of those good things in good time. So these are just some of the functional groups that you might expect to find on a very large organic molecule. Keeping in mind, the organic molecule is going to have a carbon skeleton to build around, and then these functional groups are just going to be kind of attached here and there to different parts of the skeleton until you have built your organic molecule as a whole. And whatever functional groups you have attached is going to kind of determine what the chemical properties of that molecule are. So the specific example I mentioned earlier is that if we took that heptane or octane molecule from before and we start taking off hydrogen atoms and replacing them with hydroxyl groups, the molecule would become progressively more and more hydrophilic because you're making it more and more polar. So that is going to do it for this video. I promised it would be a short one. So join us next time for the last and probably kind of a long video to end off the chapter. We will talk about each of the specific types of macromolecules one by one, starting with carbohydrates and then lipids and proteins and nucleic acids. I look forward to seeing you next time, and I will sign off for now. Bye-bye.